everyone for coming back the second day here to hear me. So this is a topic that I've been working on, and I have to acknowledge that part of this was started with Reed Johnson's input too, so thanks Reed for showing up here, and you can give me feedback later. Yeah, so as you may know, I started out as a professor at Penn State University and then transitioned 7 years ago to ARS, and has made a difference. In my lab in Logan, Utah, we have a great crew of people, and I introduced you to some of these yesterday. And just to come back and point out two people here, Dr. Kelsey Graham and Dr. Teresa Pitzinger, and myself. We've been working on asking about the impacts of pesticides on honeybees and the other bee species as well in commercial agriculture. And just to remind you, as you now know, we have a tremendous diversity of bees that are out there, 4,000 species, and probably more than that in North America. And as you fully realize, these are keystone species for agricultural ecosystems and for natural ecosystems. And so preserving them and keeping them in great health is really important. And one of the things that we've learned in the since 2006, when colony collapse disorder took place and everybody was asking, why are all the honeybee colonies dying? We know that there are many different reasons for a colony to go down. So Jeff Pettis helped coin the term, the 4 Ps, the pesticides, pathogens, poor nutrition, and parasites. And showing here that there are Venn diagrams, that they overlap with each other. These are interactive factors that each influence each other. And we now know that climate should be put on the whole thing, that it's also impacting. So talk to you today about these interactions, primarily focusing on pesticides and pathogens here. So let's talk a little bit about these other bees. So yesterday I told you about solitary bees, which each bee, the female, is like a single mom. She creates her nest, gathers pollen and nectar, brings it back into that, lays an egg. You can see the little white circles there on that tube with the brown material in it. That's the egg laid on top of the pollen and nectar. She seals it off either with soil or plant material, and then just keeps adding on new material there. That's her sole interaction with that future progeny, is laying the egg there. This is very different from what you're going to see in bumblebees and honeybees, where mom is not participating in helping to digest that pollen nectar and helping to detoxify it. And the other critical thing here is that pollen provision, that pollen and nectar combined, is in direct contact with both leaf pieces that may have come from the crop itself, or with soil that could have been there in the environment too. So this is different than what you see in honeybees as well. Bumblebees, just to remind you, it's a brand new queen. She's emerging probably from an area in the winter that is in contact with soil and goes out and starts her new nest. Often that may be in a rodent cavity somewhere in the soil too. So again, you have this contact of the colony there with the natural environment around. And then of course with honeybees, we have this resilience in numbers, where your progeny, the new larvae, are being fed by those nurse bees. And so the nurse bees, when that pollen nectar comes in, they're ingesting that pollen nectar, they're digesting it, they're also detoxifying those chemicals that might be carried over in that pollen and nectar. And then they make the protein secretions and they feed the brood. So they're a first-line defense for that whole colony. Sort of like what Sammy talked today, these progeny are getting pre-digested food, but they're also getting it where there's been a cleaning of that food, a breakdown of some of the chemicals that come in there. So you have radical differences here between solitary bees, bumblebees, and honeybees in terms of how they're being exposed. And so this is one of the things that we've been looking at. We do know that pesticides are a stressor for bees. So part of our work is to ask what that stressor is. Pesticides are a key toolbox, part of the toolbox for integrated pest management. We now say that this term should be called integrated pest and pollinator management, where you're going to control the pest that's on the crop to make sure that you can get a yield back, but do it in a way that's going to give you the healthiest bees out there to ensure that you have that pollination. So sometimes when you get exposed to pesticides, this is a slide shared by a beekeeper that was out in almonds. And the spray occurred during the middle of the day, which is not the best thing to do when your bees are out foraging. And you can see around the base of his colonies, he had a bee kill. So sometimes it's very evident that you have an impact, acute toxicity. But most of the time, you're going to see that this is at a sublethal chronic level exposure. And pesticides may be persisting in the environment and carried about by wind and rain as well. So you may have exposures coming from other sources far away. So just to remind you about some historical information here. So I was one of those people that got caught up in colony collapse disorder research at Penn State, but I also had a great team of fellow faculty members there that did some groundbreaking studies. And a lot of people, showing my age here, may not remember what happened back in 2006 or may have been younger. So this is where we had this mysterious die-off of colonies, and we mounted this major effort to try to go collect samples. So a team of Chris Mullen, Marianne Fraser, Jim Fraser, and Roger Simons helped. They took samples that Dennis Van Gilsdorp and Jeff Pettis helped gather from healthy colonies as well as those undergoing CCD and process them. So looking at the bees in the colony itself, the pollen, and the wax. They stayed away from honey for this study. And with Roger Simons, I don't think he's here at the meeting, but he is a beekeeper himself, but he was also head of the Gastonia lab for the American Market Basket Survey. They're the ones who do the analyses on food that you have off of the grocery shelves asking about pesticide inputs. And at that time, we suddenly had lost a lot of the labs that did chemical assays, and his was one of the few that we could do this on. So Chris Mullen was a toxicologist, worked on insects. He came up with a list of pesticides and worked with Roger to look at these, and basically we had this major surprise. We knew that there were pesticides out there, bees were being exposed, but we didn't know how bad it was. And this is where you had anywhere from 6 average chemicals in that pollen coming in to over 39 different active ingredients. And one thing that took them back and myself were some of these pesticides, some of these organochlorines, so like DDT, that should have gone away back when I was in junior high, but was still being detected here in 2006 in the environment. Anyway, with CCD, it was a very extensive work that they did. We could not blame CCD on any one of these chemicals or compounds or classes, but we found that they were there. And that's continued to be the case, that seems to be getting better, but there's also things that are not so great. So Kelsey Graham, I've been fortunate, we've been able to hire in the last few years. She did a postdoc with Rufus Isaacs up in Michigan, and also got an ability to work with Scott McCart, who you heard yesterday. And Scott told you about his ability to do these chemical analyses. So part of Kelsey's work that she did in Michigan is now continuing with this, is asking about what is pesticide exposure and how it's being managed. And so she looked at honeybees and bumblebees that were being brought in for blueberry pollination up in Michigan, and had an extremely good relationship with the growers that were there. So these growers were willing to share all their spray records from previous years to current, and it represented a variety of different growers, some doing very conventional ag, with pesticide-intensive control of their pests, others doing more sustainable ag, trying to be reasonable, and then some actual organic growers as well. And they collected flowers, the whole bees, the pollen, and the wax for over 2 years, and they screened with Scott's help for 291 active compounds. What they found is that both bumblebees and honeybees were being exposed to a fair number of chemicals, but they also looked at this to try to evaluate what was the risk to the bees. And so looking at the exposure, how much was coming in, and how toxic it was. And they had this data here, and there was some of these numbers that I found surprising too. So on this graph that's over on the right here, up at the top you can sort of see the first set of columns are flowers, then the bees themselves, and then pollen with that red box around it is pollen there. And there's 3 different lines on this graph here. The one in green is for the European Regulatory Agency for chronic oral exposure. The one in blue is their acute contact toxicity. It's things that when bees get it orally are usually more toxic, but acute toxicity has been the standard by which a lot of pesticide registrations were done historically. And then the line at the top is the EPA acute toxic level. And what you can see is that the pollen itself, there were 22 active ingredients detected in there, and a minimum of 12. This is coming from also those organic farms out there. And a lot of those pollen levels exceeded that European chronic oral exposure, and there were ones that exceeded EPA acute toxicity and also the European there. So this was a surprise. So asking, where did these chemicals come from? So chemicals, the active ingredients, I've now learned from interaction with many colleagues, some of them at the company that manufacture chemicals and from EPA, that label that comes with that chemical pesticide is the law, that you need to follow it to the exact level here. So some chemicals are registered for use on a crop, others should never ever be used on there whatsoever because they're not registered. And what you can see here, looking at those active ingredients and their contribution to this risk quotient, i.e. how much risk there is, that light gray part of that bar is ones that were not registered for use on blueberries, and we know from the spray records on blueberries that they were not applied. And Kelsey had extremely good relationship, felt like these fellows, maybe women too, hopefully for growers, were being very honest with her. So where did they come from? So one of the things that Kelsey did was to take that pollen incoming and ask what species did that pollen come from. And you can see that the colors here for the honeybees at the top and the bumblebees at the bottom are really quite different in color. Bumblebees like the blueberry a little bit more. We're bringing more of that back into the colonies, but you can see that they're bringing a lot of different pollens in. And the pesticides probably were coming from outside the farms themselves and probably associated with weeds. And so me, just thinking from all the literature and the popular press out there, we tend to blame corn and soybeans for being one of the culprits for off-farm exposure. Turns out this area in Michigan, the amount of corn and soybeans being grown in this area where these blueberry fields are is nearly zero. There's a lot more other fruit crops out there. There's tree farms, there's houses, and we think that this is an off-farm exposure, that these chemicals are coming from other areas away from the crop itself. So one of the take-home messages that Kelsey would like to deliver is that we need to think about pesticide exposure at the landscape level. It's not just where you're putting your bees on a particular crop, but you need to think about what's going on around. And we also need to try to educate other growers in the area that to reduce their pesticide applications outside the bloom here and look at it there. And there's a recommendation for applicators or growers that if you're have to use a pesticide, emphasis on have to use it, that you need to remove the weeds that are blooming there to prevent that exposure there and take actions, minimize drift. Okay, so shifting over to my own research. Chris Mullen, I have to give him a lot of credit. Chris has been retired now for several years, but he realized as he was doing the work on the pesticides that there was another group of agrochemicals that were not part of the active ingredient profile, and some of these were adjuvants. And there's a wide variety of different adjuvants that are out there that are used in the formulations for a lot of different reasons. And some of them are very good, put in there for a really good reason, like minimizing drift or dispersion. But there is one group here, the organosilicone spray adjuvants, that are probably there in the formulation, but they're also used in what's called tank mixing, and that's where a grower, for good reasons to minimize costs, is going to combine different active ingredients when they have to do a spray. These organosilicone spray adjuvants are not only used in agriculture, we're actually exposed to these in many different aspects of our lives. They're very popular in cosmetics. There's actually been studies done with sensors looking for chemicals and finding a spike in the early morning and another spike late in the afternoon. And it turns out hair products like the shampoos that a lot of us use have these organosilicones in them. And so they were picking up these coming off people's hairs and their bodies, leaving for work in the morning and then exiting the buildings in the evening to go home for work. So there's wide use there. It's also in industry, a lot of paints, medicine, and then I was surprised to learn in the West, manufacture of artificial snow. Hopefully these snowstorms keep us out of there, but these are widely used, and there are some of these that are of concern for medical health too, some of the cyclic forms. In agriculture, what's written in very few publications out there say that this is used for increasing spray coverage and uptake of spray solutions by the plants. So these are considered to be super spreaders, super penetrants. And then the other thing that discovered is that on the OMRI website, the Organic Materials Review Institute, where they list things for organic use, there's some of these that are received certificates. And unless a grower goes in and opens that certificate and see that it's restricted use there, they may consider these to be perfectly fine to include. So some of my colleagues who work for the agrochemical companies say that if that label on that pesticide does not say to include organosilicones in the spray application, they don't want it to be there. But I think there's another group in the industry, the middlemen. This is a way of saying include these, and it's going to make things work so much better for you. It's also a way for them to gain extra income, which I can appreciate, but they may not really fully realize what the implication is. So Chris, he went through the California database for pesticide registry and looked at all the applications of these, and Reed has continued doing this on his own right too. There's a multitude of different types of organosilicones being used, and you can see on this graph here, this is going, I think, back into the 1990s when these first appeared, and for this it's going up to 2014. That arrow that I've drawn is CCD. You can see it's an exponential increase. If you go into the California database, you see that these are not only being used in almonds, and the almond board has discouraged their use now, which is great in tank mixing, but it's also across every crop that's listed in there that they are being reported use. The other group of chemicals that these are widely used for are herbicide applications. So herbicide applications, these are put in there with the idea that you're going to get that herbicide in the plant and help kill those broadleaf plants. And Chris's work here showed that the different forms, chemical forms of these organosilicones are greatly toxic. Some of them are more toxic than others. So When I left Penn State, Chris had retired. He had developed the assays for these. I was very lucky, very fortunate at Utah State to find an environmental chemist there, Bill Doucette, who unfortunately now is also retired. But he also took Chris's assays, how he was detecting these, and improved on them. And so with funding from AHPA, Chris and his graduate student Autumn here, we did collections of pollens coming out of different crops and looked at these, and we could find that these were being used. And in many of the pollens come from almonds. This is back in 2017. We did detect these there along with other chemicals. In particular, you notice the one in red here, cyprodinol, and I probably screwed that one up, so excuse me. But it's a fungicide. It's also there with other popular fungicides. And then there's another insecticide here, chlorpyrifos, where it was high prevalence too. And some of these pesticide collections, you could tell this is probably from the same crop consultant prescribing certain applications and certain blends. But yes, indeed, these were being exposed. There were some of these also, like foxamine, which I think is used for mosquito control, was showing up too in some of these and hard to explain. So we've been doing experiments using OSS. So these are in tank mixing. If you go to the Washington State, Oregon State, IPPM website, they talk about uses there, because on the chemicals as they're sold, they're not recommending a use rate per se on their labels. Right now, these do not have any restrictions whatsoever for pollinators on there. So there's nothing on the label to restrict their use. So it ranges anywhere, I think, from 1%, 4%, 5%. On some of the turf grass labels, when I saw originally, they're saying don't use it above 5% because you're going to kill the grass. They can be phototoxic at a certain point. So right before COVID hit, like right before we got where we were told we could not travel, went out to California with my technician, and Chris Hyatt, who's an almond grower and also a beekeeper, his crop consultant had suggested or given a recommendation for an application of pesticide on his crop that was blooming, the almonds. And so he did us a favor. He applied this right before we got there or after we got there, so we were able to collect pollen before and after. And then have to thank, if Randy Oliver is here, he helped us work, yeah, Randy's here, helped us work with UC Davis up at the Nichols farm to also do an application up there. And so we were able to collect pollens immediately after application and 24 hours later. The levels were extremely high in both cases. And so we learned that, yes, indeed, what we were testing in our assays, asking about impacts, was within the field-relevant concentrations. But we also had another surprise. So a few other beekeepers that were further down in the Central Valley had not had any sprays on the crops of almonds at all. And we detected here one of the breakdown products, which sounds like a good thing, but I'm going to tell you guys it's not. See it showing up in pollen, that it was coming into the colonies at a significant amount there. So Bill Doucette, one of the things that he was famous for was asking about how plants interact with organosilicons or with, he'd done a lot of work with other type of adjuvants. And so what he set up was a hydroponic assay. So it was tomato plants, which can be easily grown in a greenhouse, grown hydroponically, and he added the OSS to the growing media. And we had control plants versus those and not. And we looked over time what happened. And just to give you the take home here, what we found was that that OSS was taken up by the plant roots and then moved throughout the plant. It went to the leaves at the highest level, but it also showed up in the flowers and the pollen and nectar. And this breakdown product, the TMS, was the highest concentrations in pollen and nectar. We think, and one of the things that got disrupted by COVID that we need to come back to, is that these breakdown products may be even more toxic than the original parent compound. And we don't know yet how these persist and carry over. But to show you some of the impacts here. So Chris had done a lot of work, so we do know that these were toxic via oral ingestion. There's a lot of work on other insects like sand fleas in this, where sand fleas, they're found in Africa. They burrow into the skin, and there's a lot of lotions and creams out there that have these organosilicones. They're used to control it. They suggested it was through just blocking spiracles. The data there now suggests that it's through ingestion of these compounds. And also that these impacted bee learning and behavior in honeybees. And with Julia, we've done work and shown that they interact with viruses, synergize with the black and queen cell virus to have higher levels of toxicity in the bee larvae. And what I'm going to tell you here is that they have impacts on queen health and survival of the colonies, and that, as Reed has shown also, they synergize with fungicides and insecticides for increased toxicity well beyond them together. And then we've also done work looking at how is this happening, looking at what genes are being impacted, and other life traits in other bees. So I told you other bees have greater exposure. We've asked about some of those questions here. So what we did, and some of you may have seen this before, we did this when I first got to Utah, we set up microcolonies and gave them the pesticides and the organosilicone in the artificial pollens, and had controlled colonies in there. And we also had marked our queens and clipped their wings so we could keep track of them, which was really good, it turned out to be, because some of those queens, when we opened up the colonies so they could go gather, we saw that workers reassorted themselves. Some of the queens must have been much more attractive, so we had colonies with a lot of workers, medium-sized workers, and extremely low levels of workers. Those queens in those colonies with the low level of workers, here the yellow and the orange bars, some of those queens, the orange bars, I came out, because this is my backyard, I would find a cluster of bees across the ground, and inside was this queen with her wings clipped, crawling across the ground, and the workers going with her, basically swarming, per se, to escape. This is brand new equipment. We also saw a lot of the colonies go queenless at this time too. And it was some of those medium-sized colonies that did this as well. So we modeled this, and what we found was that the organosilicone by itself, the pesticide and insecticide together, and then all three together, if you have a weakened colony, you're much more apt to be dead and die from that. And so having a really robust colony makes a difference. We also looked inside those workers. So some of those workers, because these are new packages, we knew that the queen had not yet had time to raise up new brood. Those workers aged over this time. So we're looking at about a 2-week sampling date here. Those bees that were feeding on the OSS, we see the synergistically higher levels of viral infections. And this is a accumulation of black and queen cell virus, sac brood virus, deformed wing virus, and Israeli acute bee paralysis virus. So these are single-stranded RNA viruses. And as you've heard here by multiple people, there's ways of transmission within the colony, but also from colony to colony via fecal-oral route, and also exposure to other bees. But these viruses went way up in those bees at a high level there. We also know that they synergize with the pesticides here, and point out that this Silgard is at 40 parts per million. This is well below what we're finding out in the field, and it makes the pesticides and the OSS much, much more toxic. We asked how these impact and how it relates back to the viruses, and we did what's called transcriptomics. We sequenced all the RNAs that are in the bee. The viruses themselves are RNA viruses, so we got sequence counts for those. And what we found was that immunity, the antiviral immunity, so there's a couple of cool genes that are called dicer, and it basically does what it sounds like. It clips these little RNAs into little tiny pieces, chop, 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 helps get rid of them. But its expression went way down. Serine protease snake, which turns on part of the immunity, was also down. And then we also saw hymnoptacin, an antimicrobial peptide, being depleted. And there were 2 cytochrome P450s. And these are ones that are normally involved in detoxification, but there is also a group of them that are essential for hormone biosynthesis, things like juvenile hormonic diazone in the bees. And JH is one of those things that are important for regulating your bee's physiology in that. We see that these were also decreased. We've also gone over to other bees. So this is the alfalfa leaf cutting bee. It's widely used in alfalfa seed production, as well as other pollinations. And this is a bee that does cut out these leaf pieces and brings it over. So we set up studies where we could do it in cages with plants where we knew the bee would like the pollen and not the leaf of that plant, and a plant that they would like the leaves and not feed on the flowers. And so we could selectively spray these different types of plants together. So, tell you the summary of what we saw, we found that bees that were exposed to the OSS, that they made fewer numbers of nests per bee. They had lower reproduction, and there were less cells in that nest as well, so that meant there were fewer eggs being laid. We also saw parasitization by a little parasitic wasp. It was about the size of a comma on a 12-point font, so it's a really little tiny one. We know that we did not bring it, somehow found a way to cages, but it liked to come into the cages where the OSS was being applied for some reason, which is another question, is this somehow making those bees much more attractive to those wasps and causing great parasitization? The kicker here, and what I like to tell my alfalfa seed growers, is that this OSS, if they choose to put it out there, spray it out there in their tank mixes, it's not only more toxic here, but they're impacting how much work they're getting out of those bees. They're impacting how well they're doing for that pollination event that those growers depend upon. This is the alfalfa seed pods here. That's what they're wanting out there. They want to get that seed yield. And if you have greatly reduced amount of pollen being brought in, that means that those bees are not visiting as many flowers out there and working for them. So they're not getting the bang for their buck from their bees that they're buying and they're trying to make work for them. So we need to ask about seed set and yield. One of my growers this year, and I heard this from others, he went off to DC or a well-deserved vacation and left his son back. And the son I know is very intelligent in this, but somebody came to him, the crop consultant, said, why don't you switch out your adjuvant for this? It's going to make things work so much better. And I need to ask him here at the end of this month, but it killed all of his bees. So here, for alfalfa seed growers, if your bees die, you basically lost your alfalfa crop too, because you can't get bees in time to replace it. And for some of these bees, the amount of bees out there is really reduced as well. We also know part of this risk of exposure that these bees, when they walk on dried filter paper that had been previously sprayed with OSS and allowed to dry, that it was toxic. And the bees, when we put them in these cages, are trying to walk on it and chew on it as well. So this other route of exposure here, this additional route of exposure, probably is real for these bees here, that they're getting this added disadvantage. We've been doing collaborations with another chemist at Utah State, Kim Hagman, and she has worked a great deal with chlorpyrifos, and we now know from work that she's done that indeed chlorpyrifos on leaves is dissipating and moving into the pollen, and they're doing work now to ask about soil and movement over into pollen as well. So these other routes of exposure for these other bees are real. So yes, this OSS is important. We need to try to ask that it at least gets a warning on there. For other pest insects, there's a lot of data out there to suggest that yes, indeed, it's a great control agent. Asian citrus psyllid, Marjorie Hoy, said that this OSS worked much better than pesticides in controlling it. It's also been shown for navel orange worm, actually, that it works as great as a pesticide too. We're doing other research, and I'll let Julia Fine at some point come back and tell you about the interactions of OSS on the queens and honeybee workers and queen health that she's been doing. And hopefully you can see this slide. So I want to get back to the idea of formulation, and this is something I think is really important for you as beekeepers to think about. And, okay. So the other thing that Chris published on in one here was comparing formulated products. So the Amitraz, which of course you guys know is a register for mite control in your bees. And there's another product out there that's used for cattle for controlling their parasites, the Tactic. One thing to show you here is that Tactic is much, much more toxic to bees than Amitraz by itself. And so formulations right now in the US, it's a trade secret. We can't tell you what's in there and what's going on. But one of the dangers, if you're going to choose a product that's off-label, potentially to use it, not that you would ever do it, right? Potentially you're exposing your bees to these other compounds that are going to make those pesticides that are coming in that pollen much more toxic and have greater impact. So I've told some of my other colleagues that are on the NAPSI Pesticide Education Task Force that I would bring this up here. And also to encourage you, we know that there's a paucity of data getting to EPA about pesticide kills. So somehow if you can go on the EPA website or work with your state agency to get those bee kills reported, we need to know when there are bee kills out there. And it may not be your fault, it's not the grower's fault, but we need to know under what conditions, environmental conditions, are these pesticides being lethal. So Tim Fredericks is here, I think, and he would be glad to chat with you about why reporting these kills are really important. Samantha Bruner's here as well from South Dakota. She can tell you about that. So I know some states like Utah, we do have very poor reporting ability going between apiary inspectors and the pesticide division. We're trying to figure out how to overcome that to get the two to communicate better. So with that, I'd like to thank everybody who helped out and to thank you for listening to me. If you have questions, go ahead and reach out. I think I'm done.
Yep, for just a second, okay?
Thanks.
Thank you. Let's stay here for just a second. Jean would like to talk to you for a moment.
Okay.
Hang around. If you can indulge me for just a minute. I'm Gene Brandy. I'm the current chairman of the Foundation for the Preservation of Honeybees. And you've probably read about Diana's bio in the program, but there's a little bit more about Diana that I'd like to let you know about. And you may not know that Diana came from a beekeeping family. Her great-grandmother was a beekeeper of high regard in Colorado, and her grandmother majored in food science and chemistry at Colorado State University. So she's from a beekeeping and scientifically oriented family. And after receiving her own degrees at Colorado State and the University of Illinois at Champaign-Urbana, Diana moved to Pennsylvania State University, where she was a professor of entomology and insect biochemistry. In the late 1990s, Diana began to look at the interactions of honeybees, varroa mites, and viruses, and she was very fascinated by the interplay of all three of these. Currently, Diana is the research leader at the USDA-ARS Pacific West Area Pollinating Insect Biology Management Systematic Research Unit in Logan, Utah. In that role, she has been involved with studies looking at flora, native bees, and honeybees, as well as the role of adjuvants in ag products, as we just saw, and potential impacts on pollinators among her many research projects. Also, the interaction of honeybees and native bees, as you heard in her previous talk, if you're fortunate enough to hear that. Very important research. And having served on this little committee with her with NAPSI, I know that she's still very involved with the NAPSI, the Pesticide Education Committee, and she's been chair of that for a while. But it certainly is an understatement to say that Dana Cox-Foster has continued her family's legacy of exploring the world as a scientist and illuminating the path into the mysteries of honeybees with the hope of providing better management of hives in the future. Her calm, reflective approach to research is a great model for beekeepers as well as researchers of all ages. So with that, the foundation would like to recognize Diana. Get this out of here. With the Founders Award in recognition of many outstanding contributions to apiculture research and education. Congratulations.
Just like you for not coming to our luncheon. We talked about our luncheon last Friday, but,
Oh, yeah. Thank you so much.
well, you're very well deserved. Thank you for all you do.
Yeah, thank you.
And you can have that. Keep that as well.
Yeah, thank you very much. This is going to go in my office and be a proud part there. Thank you so much.
Congratulations. You deserve it.
Thank you.